Hi everyone. Today we're going to talk about why hydrothermal systems are important for studying the origins and early evolution of life. So I am often asked why life had to originate in a hydrothermal system. Many people have heard about the idea that life got started in hydrothermal vents, and I have many different answers to that question, as you can see here, and that's what we're going to talk about today. So my main message for this is that I actually do not think that life necessarily started inside of a hydrothermal vent, but that during various stages in the evolution of life, hydrothermal systems would have been a really useful place for evolution. So I will explain in detail what I mean by that. The first thing I want to say, though, is that these really cool animals that we've been talking about are not reasons why hydrothermal vents are important for studying the origin of life. They're animals. Animals are by definition recent. Animals did not exist on this planet for most of the history of this planet, and they certainly weren't around 3.5 to 4 billion years ago when life first got started. So these animals are really cool. They're an important reason why I like studying hydrothermal systems, but they're recent. They are not the first organisms on the planet, and they don't really tell us that much about how life might have gotten started. Except that the reason why these animals are so cool and the reason why they live in these environments is, of course, because they have these symbiotic microbes, and the microbes that live in these animals do tell us something about how life could have gotten started. So first, let's talk about when. When did life get started, and when would life have wanted to live in a hydrothermal vent? So one somewhat controversial clue to this question is this idea of the late heavy bombardment. So the setup here is that about 4.4 billion years ago, Earth had cooled down enough. So the Earth is about 4.5 to 5 billion years old. So this is very early in Earth history. At one point, the Earth was way too hot for life to have survived on the surface of the planet. And around 4.4 billion years ago is when we think that that would have changed. So around this time, the Earth had cooled down enough to have water and maybe even oceans and maybe even continents. And we have pretty good evidence that life was on Earth by about 3.8 to 3.5 billion years ago. And there are many different reasons for that, and I won't talk about them today. Just take my word for that for now, that life had started at least by 3.5 billion years ago, if not 3.8 billion years ago. And so there's a pretty big gap between these two times when life was habitable and when we definitely have good evidence for life. And there are always problems with the fossil record, so that is certainly playing a role here, but it may not be the only reason why there's that big gap. And one clue is that there seems to have been a massive peak in the bombardment of asteroids and other large objects into the Earth around 3.9 billion years ago. And this evidence comes from looking at craters on the moon. And so by counting the craters on the moon and by dating the ages of those craters, we can get a sense of the rate of bombardments that were happening in the Earth's neighborhood of the solar system over time. And that is what this curve is showing. So on the left-hand side is 4.5 billion years ago, so the birth of the Earth. Right-hand side is 2 billion years ago, so it's going forward in time, left to right. And then the y-axis is the mass in grams of the amount of stuff that's slamming into the Earth per year. So what the curve is showing is that when the Earth was first being formed, there were, of course, many, many, many things slamming into each other. As things settled out and calmed down, the rate of bombardments goes down and down and down and down. And this is the period where Earth would have been habitable. There was liquid water on the surface. It might have been relatively normal. There would not have been any oxygen, of course, but otherwise life could have been pretty nice and during this time. And then the curve goes back up again. And this is a logarithmic scale. So this is a jump in it's like 3 to 4 orders of magnitude, so 1,000 to 10,000 times more stuff slamming into the Earth. And that's why it's called the late heavy bombardment. Late because it happens late in Earth history. This is not in the very beginning of the formation of Earth. It's happening later, heavy because there's a lot of stuff slamming into it, bombardment because things are slamming into the Earth. And so this would have been a problem for life on the surface of the planet because models suggest that there was so much stuff slamming into the Earth right during this time that the surface would have been sterilized. No microbe could have lived on the surface of the planet during the late heavy bombardment. And therefore, the only place where life might have been able to survive is at the bottom of the ocean. That's possible under a very deep ocean, in the seafloor, or maybe even underneath the seafloor. Those kinds of environments might have been protected from this massive genocide happening on the surface of the Earth. And so then the idea is that even if hydrothermal vents were not the place where life got started, they could have been a refuge, a place where life could have escaped and hidden for some period of time while the surface of the Earth calmed down and became habitable again. So that's one reason why hydrothermal systems are important. Even if it was not the place where the first organisms originated, they are very likely to have been important refugia, places where life could have hidden during the late heavy bombardment. So a related idea, but one that comes from biology instead of coming from geology, is phylogenetic evidence that thermophiles, that is, organisms that like to live in high temperatures, are our common ancestors. So there are a couple of different ways to look at this. This is one. This is a tree of life showing the three domains, archaea, bacteria, and eukarya. And the branches in this tree of life are colored by the temperature at which the organisms like to grow. So these red branches are organisms that like to live at high temperature. And what this is showing is that these high temperature, these thermophilic branches in the tree of life, which includes both archaea and bacteria, are at the root. That is, that the ancestors of all three domains, all of life on this tree, is derived from ancestors that are predicted to have lived at high temperature. So this is a phylogenetic tree. Phylogenetic trees are predictions that one needs to validate and test with experimental evidence. And that is what these authors did in one of my favorite papers. So paper published in 2003, they took a tree of life. So here's a slightly different view of the tree of life. Here are the bacteria, here are the archaea and eukarya, and they used this phylogenetic tree of ribosomal proteins. So the tree was made with proteins that are found in the ribosome, and then predicted the sequences of those ribosomal proteins in two different ancestors. One ancestor that is the ancestor of all of specifically mesophilic bacteria, so bacteria that only live at mild temperatures, and then also predicted the ribosomal protein sequences for the ancestor of all bacteria, which includes these important lineages of thermophilic bacteria, as well as mesophiles. So then what was really cool about this paper is they actually synthesized those proteins, these predicted ancestral proteins, which have not actually existed on Earth for probably billions of years. They made them in the lab and then tested their enzymatic characteristics. So this curve is showing the temperature from going from left to right in Celsius, and the y-axis is just a measure of enzymatic activity, and showing that the curve for Thermus aquaticus, which is a living thermophilic bacterium isolated from Yellowstone, it's the organism from which the first Taq polymerase was isolated, and it shows that the ribosomal proteins from this real organism alive today are, their optimal temperature is between 60 and 70 degrees Celsius. And then the curves for these synthesized proteins that they made in the lab, predicted as the ancestral proteins from that phylogenetic tree, have very similar curves as this known thermophilic bacterium, suggesting that these ancestral sequences are in fact thermophilic, that these ancestors way back when were living at higher temperatures. And this is even true of the ancestor of the mesophilic bacteria. This is shown here, is a significantly different optimal temperature compared to E. coli, which is our example of a boring mesophilic bacterium that does not grow at high temperature. The ancestor of all the mesophiles grows at a significantly higher temperature than E. coli does. So this suggests that our ancestral ribosomes, the ancestors of all bacteria at least, were optimized for functioning at high temperatures. Experimental validation of this observation from the tree of life. So here's a more recent study that was inspired by that one, and this one it does not have experimental laboratory evidence. This was only a simulation, but the results of the simulation were still pretty interesting. So once again, we have a tree of life. Here are the bacteria, here are the eukaryotes, here are the archaea, and they've color-coded their tree by temperature, by growth temperature, the optimal temperature that these organisms like to live. The eukaryotes are grayed out because their sequences weren't really compatible with the simulation, and anyway, eukaryotes don't ever live at high temperature, so it wasn't interesting to include them. These numbers here are in degrees Celsius, the predicted temperatures that the ancestors at these positions in the tree grew at. And if you look at the ancestors of the archaea and the ancestor of the bacteria, consistent with the previous study, they predict a high temperature for these ancestors. These ancestors of the bacteria and the ancestor of the archaea would have grown at high temperatures. Okay, that's nothing new. That's what we saw before. But then when they predicted the growth temperature of the ancestor of all life, of all three domains, bacteria, archaea, and eukarya, they actually came up with a mesophilic prediction. That is, that their prediction for the last universal common ancestor of all life grew at a moderate temperature. And this was a surprising result, right? If you just averaged the predictions for the bacterial ancestor and the archaeal ancestor, you would have ended up with something between 50 and 60 degrees Celsius, right? But no, they came up with a prediction of 20 degrees Celsius, and if you want to know more about why they came up with that surprising result, I recommend this paper. They go into the molecular details of that. But what I think is interesting is that this is very compatible with this idea of the late heavy bombardment, right? If Earth got started at a normal temperature, maybe in the surface of the ocean or in a warm pond somewhere, the late heavy bombardment occurred, all life on the surface of the Earth went extinct. The only life that survived on the planet was living near hydrothermal vents and the warm seafloor, and it would have been warm anyway because the surface of the planet was boiling. And so the only organisms that could survive could tolerate high temperatures, maybe even prefer to grow at high temperatures, and they are the ancestors that gave rise to all of the life that is alive today. So that's just one story. It's a fun story. It's a story that I like, but it's just one prediction, and we need to study this a lot more to be more confident of it. But those are two reasons so far that we've given for why hydrothermal systems are important for thinking about the early evolution of life. Let's move on to number 3, which is the synthesis of organic molecules. So you have probably heard of the Miller-Urey experiment before. It's one of the most famous experiments in science. It was inspired by early ideas about the composition of the early Earth atmosphere, which is that it would have been very reducing. That is, it would have had things like hydrogen gas and methane and ammonia. There would have been lightning. And so Stanley Miller mixed these things in a lab with an electrical spark, which is approximating lightning, mixed them together over a number of days and collected the brown gooey stuff that came out of the results of this kind of mad scientist experiment, and characterized the gooey tar that came out of it and found lots of amino acids, and as well as other stuff that had formed during this experiment. And this is often pointed to as the experiment to show that biological molecules like amino acids can be produced from non-biological molecules like hydrogen gas and ammonia and methane. It was a very influential experiment. I think it's also interesting to point out that you can get these same amino acids in meteorites. And people have done a lot of work to prove that these meteorites really do come from outer space and are not just contamination. And the types of amino acids, including both biological and non-biological amino acids that you find in meteorites, this Murchison meteorite is the most scientifically useful one that so far, as well as this discharge experiment. This is the Miller-Urey experiment. They're very similar. You get very similar and very similar amounts of both biological and non-biological amino acids in both meteorites and the Miller-Urey experiment. So what this means to me is that it's just not difficult to make amino acids. Amino acids are everywhere. They're literally raining down from the skies. And to me, what the really important question is, how do you synthesize complex organic molecules from these simple precursors? So how do you take single amino acids and make them into a protein or an enzyme? How do you take single nucleotides and polymerize them into DNA and RNA? So we have some clues to that question from hydrothermal settings. So here is a laboratory simulation of hydrothermal settings. So in this study, they reacted formate, which is a very simple organic molecule that is predicted to be formed in hydrothermal systems, powdered iron as an approximation of the kinds of minerals that you would find in the rocks of hydrothermal systems at a fairly typical hydrothermal circulation temperature of 250 degrees Celsius, which is of course way too hot for life, but it's the right temperature where a lot of chemistry occurs in hydrothermal systems and under the kind of pressure you would see in these systems. And in the end, you generate hydrocarbons. So you polymerize carbon, you make carbon-carbon bonds under these kinds of hydrothermal systems. This is a study from the Lost City system that I talked about in a previous lecture, looking at hydrocarbons, so carbon polymers in these natural chimneys. So this is not a laboratory simulation anymore. This is actually going out to the chimneys on the seafloor, collecting samples of the fluids venting from the chimneys and measuring the gases and other chemicals inside the fluids venting from the chimneys. And this shows that single carbon hydrocarbons are quite abundant, and as you look at molecules with 2 carbons or 3 carbons or 4 carbons, the concentration goes down, but you can still detect even 4 carbon molecules in these chimneys, suggesting that these hydrothermal chemistry occurring in the subsurface is actually generating up to 4 carbon molecules and probably even more. That's number 3, synthesis of organic molecules in hydrothermal systems, which directly relates to my reason number 4, which is that chimneys act as natural PCR machines. So let me tell you what that means. So we just saw that hydrothermal systems can actually make carbon-carbon bonds and make larger organic molecules. But that still doesn't tell you how you get things like DNA. And so that's what this study showed. This was a simulation. I'll show you the experimental validation on the next slide. But in this one, this was a simulation inspired by images of lost city chimneys. And they were looking at the structures of these chimneys. So this is a microscope image of a chimney that's been sliced open. So there's a thin section. And they're looking at this interesting organic-like structure of the minerals forming these chimneys. And they noticed that the structure of these kind of micropores, these fractures within the chimney structure, coupled with the very strong thermal gradients that we talked about in our previous lecture. So you can have very high temperature and very cold temperatures that create convection currents inside these micropores. So thermal gradients and micropores and these microchambers, that is basically what a PCR machine is. You're cycling a thermal gradient in a very small volume of liquid, and they predicted that's what these chimneys would actually do in nature, is create a thermal cycling system. And then they validated this in the lab. So they constructed a hydrothermal chimney simulator. They created a heat flux going through their simulated chimney, which created a convection system inside the micropores. They inserted some DNA molecules of this size distribution. The second lane of this gel is what came out the other side of the chimney. And then the third lane are the sizes of the DNA molecules that stayed inside the chimney. So what this is showing is that when you cycle DNA molecules through a hydrothermal chimney in the lab, because of the convection current generated inside the hydrothermal chimney, larger molecules are preferentially trapped inside the chimney. And then the idea is that those undergo further polymerization. The larger molecules are further selectively enriched, which then can undergo more polymerization. And this is how you can imagine nucleotides polymerizing themselves into large DNA molecules in nature, inside the pores of a hydrothermal chimney. So that's number 4. So not only can you make simple organic molecules inside of a hydrothermal system, but once you have simple organic molecules, you can then polymerize them inside a thermal gradient inside the chimneys. So these are obviously very early studies, and we do not have definitive proof that this is actually what happens, but they're very intriguing early studies that indicate that there's something interesting going on. All right, my last answer for why hydrothermal systems are important for studying the origin of life comes from further observations of the needs of life that we can study today, as well as from geochemistry. So let me explain what that means. So first, I wanted to show you this slide because I should have shown it in a previous lecture because it's pretty awesome. I highly recommend this review, which I have posted on Canvas. And this is an overview of all of the many different kinds of autotrophic systems found in the ocean, and specifically the deep ocean, and highlighting especially hydrothermal vents here. So these yellow columns are electron donors used by different metabolic strategies and by microbes in hydrothermal systems. The blue columns are electron acceptors. So of course you see things like oxygen and nitrate, sulfate and sulfur and iron. So these are different electron donor acceptor couples that you can find inside hydrothermal vents. These are the types of organisms that use those strategies. And these abbreviations are the different kinds of carbon fixation pathways that are powered by these donor acceptor couples. So these are all autotrophic strategies, all carbon fixation pathways that are fueled by these combinations of electron donors and acceptors. So what I want to talk about is one of these. This WL is an abbreviation for Wood-Lundahl. The Wood-Lundahl is a carbon fixation pathway, just like the Calvin-Benson cycle. It fixes carbon dioxide into biomass, but it does so only with hydrogen gas as electron donor and carbon dioxide as its electron acceptor. And people have been really interested in the Wood-Lundahl pathway for a long time because it has many elegant features of it that just scream out the possibility that this might have been the first carbon fixation pathway. One of them is that the electron acceptor, carbon dioxide, is also the thing that you're fixing. So you're fixing carbon dioxide by using carbon dioxide as your electron acceptor, which is just kind of wild when you think about it, and just very elegant. The second thing that's elegant about it is that normally we think of carbon fixation as a process that requires an input of energy, right? You need to generate ATP, and then you spend that ATP in order to fix carbon dioxide. Well, the Wood-Lundahl pathway is a carbon fixation pathway that is also an ATP generation pathway. It actually conserves energy while it is fixing carbon dioxide, so it actually has a net gain of ATP. It does require inputs of ATP in key steps of the carbon fixation pathway, but overall, it generates more ATP than it consumes, which is another very elegant aspect of it that really screams out that this could have been the first carbon fixation pathway and maybe even the first important metabolic pathway. So who uses this really interesting carbon fixation pathway? This is only used by methanogens and acetogens. So all known methanogens are archaea. They are the only organisms that can make methane, and they do so by using this Wood-Lundahl pathway, fixing carbon dioxide with methane as the waste product. And there are bacteria that use the Wood-Lundahl pathway, but those bacteria are acetogens, not methanogens. They're called acetogens because they use the same pathway, but instead of making methane as their waste product, they only make acetate as their waste product. And this is one of those biological mysteries that there is no good explanation for why archaea would only have methane as their waste product and bacteria would only have acetate as their waste product. There's just no reason why that has to be so. And when you find biological observations like that, where there's no reason why physics and chemistry say it has to be this way, there must be a really important biological clue there, perhaps even a big clue into the divergence of bacteria and archaea from each other in the first place. So that is a mystery. We don't actually know any more than that, but it really seems like an important observation that will someday lead to a big discovery, is my prediction. Okay, but today what I want to point out is that this Wood-Lundahl pathway used by methanogens and acetogens requires many different metal cofactors. So that is what is being highlighted in red here. This first step requires molecules that use iron sulfur as cofactors, tungsten, selenium. This step requires zinc and iron sulfur, another zinc step. This step requires cobalt. This step requires a nickel iron sulfur cofactor. This has sulfur as an important component of the molecule. This last step that makes acetate requires manganese. So you have all these different metals in one metabolic pathway that might have been the very first metabolic pathway. And so this just screams out that this metabolic pathway must have evolved in an environment where you had an abundant source of dissolved metals. What kind of environment is that? Hydrothermal systems are the only systems where you have lots of dissolved metals around. Here are two more papers to show that you can get some of these steps in the Wood-Lyngdal pathway to go in the lab without biology. So in both of these papers, they took some inorganic molecules in a sterile environment and got them to produce acetate in this case and pyruvate in this case, so important biological molecules in carbon fixation. And in this step, the reaction would only happen if you had iron sulfur, nickel sulfur, and selenium available as catalysts. And this one was only catalyzed by iron sulfur. So this is exactly what you would predict just by looking at the requirements of this metabolic pathway, is that perhaps this metabolic pathway evolved from an already existing geochemical system, where geochemicals, without the help of biology, were already making organic molecules, and then life evolved as something that was taking advantage of this chemistry that was already occurring. And of course, in an environment where it was suitable for it to occur, like a hydrothermal system with lots of metals flowing by and at relatively high temperatures. So I'm showing this slide to say that even if this did not happen in a hydrothermal vent, this requirement of metals, especially in biology, is a really important clue into how life must have evolved. If it was not in a hydrothermal system, it had to have been somewhere else where there was a good source of metals, because it's not only methanogens and acetogens and extremophiles and hydrothermal systems that have these metal requirements. All life on the planet requires many different metals for them to survive. So in yellow are the elements that we normally think of as being the elements required for life. CHNOPs, you might have heard of before. Green ones are the trace elements, we often call them, because you don't usually need them in very high concentrations, but you definitely do need them. These are believed to be essential for all bacteria, plants, and animals. This is why we have vitamins, for example. Many of our vitamins are really just organic molecules that bring along metals with them, iron and nickel, and copper and zinc, for example. And so, if it wasn't hydrothermal vents, it had to be somewhere else where you could get these metals to evolve these metabolic pathways. So all of these reasons that I've been talking about, and many more, have inspired many people to think about hydrothermal vents indeed as the place where life got started. I just give you many reasons why, even if it did not start in hydrothermal vents, it was still an important place for different steps in the evolution of life. But there are people who have, in fact, proposed that it did start in a hydrothermal chimney, and here's one very elaborate proposal by a guy named Mike Russell, who's been very passionate about advocating for hydrothermal chimneys as the place where life must have gotten started. He thinks of the pores inside hydrothermal chimneys as the very first protocells. Cell membranes would have formed around the foundation of this geochemical structure of the chimneys, and he even envisions multiple different steps in the evolution of life, all happening inside these chimneys until you finally have free-living bacteria and archaea who emerged from these chimneys. So those are the 5 reasons that I often talk about when explaining to people why I think hydrothermal systems are really important for considering in the early evolution of life. But I always like to point out that there's no reason why hydrothermal systems were the only systems that were important for the origin of life. We know today that life is a global phenomenon. We often talk about the global ecology of life on Earth. Even if you don't subscribe to Gaia, it's clear that life is connected globally, that all the ecosystems on the planet are connected with each other in really important ways. And I think there's no reason why the origin of life would have been any different. I think of the origin of life also as a global process where chemical and physical processes all over the Earth were working together, if you want to anthropomorphize it that way, to form life. And life is really complicated, has many different requirements, and it probably took an entire planet of of different geochemical and physical processes to make it. So here's a review that I wrote with a bunch of co-authors when I was in graduate school, and where we put forward this idea that life originated from a global chemical reactor and not in only one setting. I think hydrothermal systems are a really important aspect of that picture, but I would never say that they are the only place where life got started. All right, that's my lecture on hydrothermal systems and the origin of life. I hope you liked it. I'll see you next time.